Hello students, welcome to Vedantu CBSE NCERT Solutions. So today we are going to do our sexual reproduction part 3. So we were continuing, and yesterday we did part 2 where we understood up to post-fertilization events or starting of post-fertilization events, and yesterday we did the outbreeding device, inbreeding depression, all these topics. Today we are going to complete from there, and we are going to complete the chapter as well. So let's directly dive into the video. Before that, let me also check if I am live and audible at the same time. Yes, we are live. Depression, all these topics. From there, and we're going to complete the chapter as well. So very nice. We are live and we are audible as well. So yes, yesterday's class we did outbreeding. One second. Yeah, outbreeding devices. So all of you can tell me what is outbreeding devices, or why do we use outbreeding devices in the chat? Okay, what is outbreeding devices and why do we use outbreeding devices? And you can just tell me one word or just a statement answer about it. Yes. Hi, good morning to all of you. And those students who are new to the class, do not forget to subscribe to the channel and also smash the like button, okay, because this class is going to be very, very important. And yes, what is the use, students? So the use of outbreeding device is that whenever there is inbreeding depression, that is a plant goes through continuous self-pollination, okay, when the plant is going through continuous self-pollination, the plant will go through something called as inbreeding depression, where the plant will get tired of doing self-pollination and there is no variation, okay. That is when we'll be using outbreeding devices. So yesterday we saw which are the outbreeding devices. First thing, the plant itself, okay. So the plant itself, students, without taking anyone's help, the plant itself will make methods telling that I don't want to do self-pollination, so I am going to use this technique for that instead, okay. So the things that plants do is it will make sure that it makes unisexual flowers, okay. So because there is chances of self-pollination, because both the plants, right, both the plants or both the stigma as well as the anther is available, that is the reason why self-pollination happens. So the plant will be like, wait a minute, I do not want self-pollination to happen, so what I'm going to do is I'm going to make unisexual flowers. First method of that. Second thing that they're going to do is they're going to change the position of the stigma and the anther, okay. They're going to change the position of stigma and anther, that is where the stigma will be above and the anther will be below. Now if the anther, okay, if the anther has to dust the pollen grains on the stigma, then what is the thing? Position have to be changed, so if the position is changed, then the anther will not be able to dust the pollen grains on the stigma, okay. That is the second thing. Third thing is self-incompatibility, that is when the stigma, so it is very simple, students, when the pollen grains falls on the stigma, as I told you, wind, water, bees or any agents for that matter are not going to select the pollen grains telling that okay, this might be right for this fellow, so I'll take this pollen grain and go. No, they are going to just casually take it and go. No, so there are chances that there is right pollen, there is wrong pollen. But whenever the self pollen, okay, that means from the same plant on the same flower the pollen is falling, the stigma will become incompatible, okay. It will become incompatible. This way what will happen? Good morning Amrit, good morning Dikshi. What is going to happen? Immediately the rejection is going to happen. This way self-pollination can be avoided. Then the fourth method which we studied was what, students? That is when the anther and the stigma's receptivity, that is when they mature. Anther will mature faster, stigma will mature later. That time also self-pollination cannot happen. Same way, vice versa. So this way they were able to avoid inbreeding depression, or the plant by itself was able to avoid inbreeding depression, or else we can also do different techniques. Right, we as humans can do artificial hybridization if we want a certain type of pollen grains to land on the stigma. So that kind of modification can be done. Then people started telling that okay, wait a minute, autogamy cannot, like autogamy can be avoided, but geitonogamy cannot be avoided because of this, and that is the statement here mentioned. Okay, geitonogamy is where from the same plant two different flowers are going to mismatch. Okay, pollen grains are going to fall from the male flower to the female flower on the same plant, but they're different flowers. Okay, geitonogamy. But understand, geitonogamy is like marrying your Mama Purna, we tell, no, in Tamil and all, Mama Purna. So how much of variation can ever happen? Very less variation, no? I think all of you know in your villages and all, if they marry Mama Purna, that means they are belonging to the same family, right? So that's all. That is like ethnogamy. You're not you're not marrying someone from your own bloodstream, but you're marrying the same bloodstream another person. Okay, so that much only variation will happen. Still, the diseases, the genetic disorders will still continue in the same blood. Okay. So that is what they're telling. Then students, we understood about pollen pistil interaction. So yesterday I could not, go in detail about it. So today I'll tell you about it. Pollen pistil interaction is the technique where the stigma will get to choose. Right? The stigma will make the choice. Stigma will make the choice. Now stigma will make the choice means what? Stigma is going to only allow the compactable, okay, compactable pollen to form or to grow the pollen tube. To grow the pollen tube. Now what are you trying to say? What are you trying to say? The thing is that, students, if let's say this is the stigma. Okay, this is the, okay, this is a beautiful stigma. On this, let's say one pollen fell. Okay, so let's say Amrit was wind pollinated. Amrit is the wind. Okay, Tanuja is water. Okay, and we have Soni B is here. So instead of Soni B, we have Dikshi B. So these three people have landed 3 pollen grains on the stigma. Now, these people did not know. Amrit, Tanuja and Dikshi did not know what pollen they are carrying with them. They just knew that, okay, they are carrying the pollen and they have dusted somewhere. Now, the stigma knows exactly which pollen is going to be compatible for them, and that is going to be done. The compatibility is going to be checked with the help of chemical dialogues. Students, they are going to exchange dialogues, like how we talk, no? If a bank manager comes to get married to you, what will the bank manager tell? I will give you how much of a loan you want. Will they tell like that? I don't think they'll tell like that. They'll be like, don't worry, I get a lot of loans, we can travel abroad and all that. They'll tell. If a doctor is coming, the doctor will tell, I will always take care of your health. I will not have time to spend with you, but I will always take care of your health. I will take care of your family's health. I'll make sure your parents are healthy. Then if an engineer comes or if some other business person comes, he'll be like, don't worry, we have a lot of money. We can travel abroad. All these they will tell. Right, all these things they'll tell. So, like that chemical dialogues will happen between the pollen. It is not like bribing, okay, telling the pollen that, wait a minute, if you make me grow the pollen tube, I'll give you 50,000 rupees. It's not bribing, but it's a chemical dialogue between 2 of them, so they know who is compatible. So once the chemical dialogue happens, okay, let's say the one that was bought by the wind, okay, wind pollination started to grow pollen tube. That means what? The one that Amrit bought, okay, the one that Amrit bought started growing into pollen tube because they are compatible. Okay, they are compatible. Are you all listening? Yes or no? No response I can see. Okay, they are becoming compatible. Pollen tube germination has happened. That is what is called as pollen pistil interaction. Pollen and the pistil will interact with each other with the help of chemical dialogues, and then the stigma will understand that, okay, pollen pistil means stigma is a part of it, right? So stigma will understand that, okay, wait a minute, this pollen grain is going to be compatible, so I'm going to let this pollen grain grow the pollen tube. Understood, students? Okay, understood? So Amrit has successfully passed the test of compatibility. Okay, so yes. So often pollens of the wrong type, neither of the other species or from the same plant itself. So same plant, okay. Let's say the one Tanuja bought was from the same plant, but because of inbreeding depression, this will get rejected. Why this will get rejected? Because the plant had already decided that I'm not going to do self-incompatibility, right? I'm already going to do self-incompatibility. That is, I'm not going to accept my own pollen. So that is why this one got rejected, okay? And this one was the wrong type. This one was the wrong type. So, neither from other species or from the same plant, if it is incompatible, also lands on the stigma. The pistil has the ability to recognize the pollen, whether it is the right type or the wrong type, okay? If it is the right type, pistil accepts the pollen and promotes post-pollination events. Clear? Now here you can see, here they have already spoken about the events, but later in the chapter only they are talking about double fertilization. So I will go in flow of that to make you understand. Students, can you see here? These are the pollen grains that has fallen. Can you see this thread-like structure? These are the pollen tube germination. Okay, it can grow. Now this one is very silky kind of structure, but if you see it under the microscope, you will be able to see like this. Okay, you will be able to see like this. This is how pollen tube germination will happen. Okay, like this growth will happen. One might be smaller, one might be bigger. Okay, it will take its own time and it will start growing. So here you can see right and wrong ones are there. It is going. This image we'll come back to. Okay, with this image we'll come back to. Now look here. If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination to the stigma or the pollen tube growth. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of continuous dialogues between the pollen grain and the pistil. Okay, it exactly looks like a proper marriage thing, no? That okay, I will reject you, I am not compatible with you because the way you spoke I did not like, blah blah blah. Okay, so like that only this conversation is going to happen. So the blah 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 between the pollen and pistil is called chemical interactions. Okay, so even though I'm telling it very funny, remember that okay, ma'am told some blah blah blah, it is a chemical interaction. Got it? Now, it is only recent years that the botanist has been able to identify some of the pollen pistil components and interactions leading to the recognition followed by its acceptance or rejection. So, in very recent time only, they got to know that actually the stigma decides which pollen is good for me or which pollen is not good for me, or right or wrong type, and then it leads to acceptance and rejections. Okay, clear, students? Now, pollen tube grows. Okay, pollen tube grows through the tissues of the stigma and the style and reaches the ovary. You would recall that in some plants pollen grains are shed at the two-celled condition. Two-celled condition. What is two-celled condition? I had already explained all of you. So students, they are trying to say in most 60% of the angiosperm, two-celled condition means one vegetative cell plus one generative cell. One generative cell means one male gamete. Okay, the generative cell is going to carry the male gamete. This also I hope you all of you know. So we call the pollen grains as the gametophyte who is going to carry the gamete. Okay, one male gamete. But the rest 40%, again this is very vague number. Okay, 40% is going to do it in the three cell stage. That is one vegetative cell plus two generative cell. Two generative cell means I'm talking about two sperm cells. Okay, two sperm cells or two gametes, male gametes. Okay, two gametes. Now you tell me, for marriage, okay, let's say it's your marriage, okay, you are going to see the bride, groom. So there is doctor, engineer, banking person, but there is two people who has come. Imagine your marriage is going to happen and you expected only one person, the elder brother, to come and see you, but with the elder brother, younger brother also came. So now what are we going to supposed to do? What are we going to do? Like he also came and he's like, I also want to get married. I got to know that you have a sister or you have a brother. So we have to get married together at the same time and all that. So suddenly what will happen? Instead of double fertilization, triple fusion will happen, right? Triple fusion will happen. So same way here also it happens, students. In the two-cell condition, only one marriage is going to happen. That is your marriage, okay? The actual bride and the actual groom. But in second situation, when it is three-cell stage, when the generator cell is bringing two male gametes, which was unexpected, one of the male gamete is going to fuse with the polar nuclei. So that is when triple fusion happen. And why is it called triple fusion? Because two people are there, no, two cells are there. Polar nuclei is two cells. So those two cells also will fuse with this one person. Understood? So that is where triple fusion word will come. Is this clear? I'm connecting with daily life so that it will be easy for you to remember. Okay? Now don't think why is this ma'am so much talking about marriage. No, it is actually easy to remember. Now look here, the generator cell divides and forms two main gametes during the growth of the pollen tube in the stigma. In plants which shed pollen in the three-celled condition, pollen tube carries two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. So I told you filiform apparatus is the person who will be like, come inside, he'll call, no? So this pollen tube will enter through the filiform apparatus to the synergid. After reaching the synergid, the main marriage is going to happen. No, that is going to be between the egg and one of the male gamete. Okay, that will form a zygote. But one more person came, no, without telling us. That fellow will go and get married with the polar nuclei, okay, or their fusion will happen. Because they are going to be three-cell combination, it is going to be triple fusion. Clear? Now, many recent studies have shown that the filiform apparatus present at the micropylar end of the synergid guides the entry of pollen tube. Okay, All these events from the pollen deposition on the stigma until the pollen tubes enter the ovule are referred to as pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help in plant breeders in manipulating the pollen pistil interaction even in incompatible pollination to get desired hybrids. Okay. Now, shall I teach you pollen pistil interaction? Shall I teach you double fertilization here only? Because later in the class also it is there. So, very simple. First, let's make it like one story only. Okay, this is the stigma, very bad stigma. 1 second. I care a lot about how it looks, students. Okay, stigma is here. Now one stigma will draw here, one stigma will draw here. Okay. Yes. Now on the stigma, we already understood one person is coming here, one pollen came here. Let's say it's someone, okay, some doctor, engineer, bank manager, business person, someone and all came. Okay, someone and all came. Now among this, let's say pollen pistil interaction happens. The first ever step is going to be, okay, first ever step is going to be what? Chemical dialogues. Chemical dialogues. This chemical dialogues, a chemical interaction, let them know that who is the perfect match for them. Okay, so now let's say the pollen has started growing, pollen tube growth has happened. Now the pollen tube has grown, students. Now I'm going to draw the same image here. Okay, pollen tube was there, it started growing all the way up to here. Okay, oh no, I cannot go back. Okay, let's draw like this. Okay, it went all the way down. Okay, all the way down, and it stopped here, and it's continuing like this. Now when the pollen tube is growing, remember that they are going to bring all the content also with them, right? They are going to bring all the content also with them. Now let's say, students, here your embryo sac is there. Okay, here your entire embryo sac is there. In that embryo sac, okay, in the embryo sac, here your synergids are going to be there. Okay, synergid 1, synergid 2. Okay, other things I'm not going to draw because it will become too clumsy. Here the egg is there. Okay, so this synergids will have filiform apparatus, Now in the pollen tube, what and all do you expect? All of you tell me in the chats. What do you expect in the pollen tube? Tell me quickly. In the pollen tube, you are going to see one generative cell, male gamete, okay, another generative cell, male gamete, and the vegetative cell. Now remember that by the time the pollen tube reaches half, the vegetative cell will completely disappear. Okay, vegetative cell will degenerate. Why it will degenerate? Because it has given all the content, all the nourishment to the growing generative cell or the male gamete. Okay, so it had only content for one, but since it became 3 cell state, it has given all the content and it has got completely over, right? I'm giving continuously food to you, I've become poor. So like that means what? I will completely degenerate. So here by the time it reaches here, vegetative cell has got degenerated. Now look here, students, this male gamete and this male gamete, both of them will enter the synergid, one of the synergids, and here one will go and fuse with the egg. Okay, so let's write it here. Second step of your this one is pollen tube germination. Okay, second step is going to be pollen tube germination. Second step is going to be pollen tube germination. First step of this chemical dialogue. Then this pollen tube germination will continue, and the third step will be where it enters. Okay, many steps you can write actually that pollen tube germination is there, the content is coming. Third step is going to be where the pollen tube is meeting the girl. Okay, the groom and the bride is going to meet each other. So here what is going to happen? It will be the entry of pollen tube, entry of pollen tube through filiform apparatus. Okay, through filiform apparatus. Clear or not? All of you? Yes, through the filiform apparatus. Now they are going to enter the synergids. Okay, after they enter the synergid, students, after they enter the synergid, the next step is going to be the one male gamete. Okay, one male gamete is going to fuse with the egg. It is going to fuse with the egg, and this will form a diploid cell called as zygote. Diploid cell called as the zygote. Now one more boy came, no? Now we accepted only this fellow. We accepted only this fellow, but this fellow is like, no, even I want to get married, even I am a very nice person, blah blah blah. But that is not the case exactly. What is going to happen in this only, the generative cell has split. Okay, generative cell has split to form one more male gamete. Now that one more male gamete has to fuse, no? So that will go and fuse with the polar nuclei here. Polar nuclei here. So the second scenario is going to be one male gamete. Okay, one male gamete going to fuse with the polar nuclei. Okay, polar nuclei or the in the central cell to form. Excuse me, started. Okay, 1 second. So sorry, students. Okay, one male gamete is going to fuse with the polar nuclei to form what? It is going to form pen. Okay, that is excuse me. Yes, is this clear? Primary endosperm nucleus. Okay, primary endosperm nucleus. This is going to be a 3N. This is going to be a 3N. Clear, all of you? Yes. Clear, students? Yes, okay. So this is what is going to happen. Okay, now when we go further, you will see from NCERT. So students, NCERT, even though they do pollen pistil interaction, then later only they're talking about double fertilization. So you might have a confusion. Okay, so what we will do is I will come to double fertilization, then I'll come to emasculation and bagging because they're not connected. So look here, double fertilization, what I just told you, try to remember. Okay, after entering one of the synergids, the pollen tube releases two male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing syngamy. So syngamy is going to happen with the initial marriage, okay, the actual decided marriage where one male gamete will be there. One male gamete will be there that will fuse with the egg. Because egg and the male gamete is the important part. No, egg is the main female gametophyte. So that is what is important, female gamete. Okay. So one male gamete with the egg will cause syngamy. Syngamy. And what will they give rise to? They are going to give rise to a diploid cell. Got it, all of you? Yes. Good morning. Students, all of you mark your attendance, okay? Click that like button, all confused people. Now look here, thus completing syngamy. Now this results in the formation of a diploid cell called the zygote. Now the other male gamete moves towards the two polar nuclei. So two male gametes which was given by the generative cell, always remember this only happens in a 3-celled stage. Okay, 3-cell stage where there is two generative cell and one vegetative cell. So in the 3-cell stage, one more male gamete was released. No, that male gamete, what it will do? That will move, okay? That will move one male gamete. Students, today biotechnology chapter is there. Don't forget, just inform your friends as well. Okay. Okay, one male gamete, students. It is going to fuse with the polar nuclei. And together form what? They are going to form primary endosperm nuclei. Primary endosperm nuclei or the pen. Okay, pen. Primary endosperm nuclei or the pen. This is going to be what? This is going to be triploid. This is going to be triploid. Yes or no? This is going to be triploid. Got it? So one diploid cell has been formed, one triploid cell has been formed. This is called as pen. Now look here. The other male gametes move towards the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus. As this moves, the fusion of three haploid nuclei is termed as triple fusion. Since two types of fusion, that is syngamy and triple fusion, takes place in an embryo sac, the phenomena is termed as double fertilization. So students, if two people's marriage is happening in the same home, we tell no, oh, two people have got married, right? So it is a double marriage. So same way, double fertilization. One haploid cell has been, one diploid cell has been formed, one triploid cell has been formed, one zygote has been formed, one endosperm has been formed, and this endosperm is going to give all the nourishment for the growing zygote. Yes or no, clear, all of you? Yes, done, done. Okay, okay, done. Now, this primary endosperm nucleus and even unique to flowering plants, the central cell after triple fusion becomes the primary endosperm cell, that is the PEC, and develops into endosperm while the zygote develops into an embryo. So the fate of these people, what is the fate of these people? Pen, okay, that is primary endosperm nucleus, the fate of it is going to become an endosperm. Okay, endosperm. And the fate of the zygote going to become the actual embryo. Okay, actual embryo. Done. Everything is clear? I'll write a box which is given in Tattva, okay, for all of you. So just copy this down with me. This is, students, this is very important. Write it next to your NCERT only. Don't be careless. Don't be lazy doing that. No need, because this notes you're not going to get. Okay, this notes you're not going to get. I'm not going to share these notes because these are written using these pen. No, they will not be eligible to read. So right now only write it. Next to your NCERT only you write. You don't have to write it anywhere else. Okay, double fertilization is equal to, okay, syngamy plus triple fusion. Okay, triple fusion. Syngamy plus triple fusion. Now here I'm going to write like how they are going to be. Okay, so syngamy is going to be one male gamete plus female gamete egg going to form, one second student, yeah, going to form zygote, and the fate of the zygote, this is going to be 2n. Okay, the fate of the zygote is to form the embryo. Okay, this is syngamy. Then when I'm writing triple fusion, one male gamete, okay, plus 10. Okay, one male gamete plus 2 polar nuclei is going to form primary endosperm nucleus. That is going to be 3N. The fate of this is going to be endosperm. Clear? There cannot be a better flowchart for explaining this. This is the best flowchart possible. Okay, I'm just going to check if it is visible for all of you. Visible, no? Clarity is there, right? Yeah, okay, visible. Yes, this is the best flowchart possible. Just write it next to your NCERT and you will thank me later when your classes, like when your exams are going to come. Okay, when your exams are going to come, you guys are going to thank me. So is the process clear? Double fertilization is clear. Usually people complicate this, but you don't have to complicate so much. There is nothing so much to complicate. Okay, it is very simple. Very, very simple. Two male gametes are there. One is fusing with the actual egg, one is fusing with the polar nuclei, and one is going to form the endosperm, and that endosperm is going to give all the nourishment for the growing embryo. Okay, because even in embryo sac we saw nucellus was giving all the nourishment. In anther we saw tapetum was giving all the nourishment. Same way, someone should be there to provide the nourishment for the growing, let's say, gamete or the embryo. So here it is going to be the endosperm. Got it, students? Chalo. Okay. Okay, now I'll come to this one. Okay, 1 second. Let me also check here. Yeah, now I will come to artificial hybridization. Okay, so see, you can easily study pollen germination by dusting some pollens from flowers such as pea, chickpea, balsam, vinca rosea on the glass slide containing a drop of sugar solution. After 15 to 30 minutes, observe the slide under the low power lens of the microscope and you will see pollen tube. Okay, this we have done in college. This is actually what you do it in college. PU, you would have done it. I don't know if you guys have done it. If you have done it, just put a thumbs up in the chat. So we usually do this and you will be able to see the pollen tube growth. Okay, now as you shall learn in chapter on plant breeding, a breeder is interested in crossing different species and often genders to combine desirable characters to produce commercially, very important word, commercially superior varieties. Okay, commercially superior varieties. That means what, students? That is, if I want to make, okay, a combination, let's say today biotechnology I'm teaching, so we are going to talk about the similar thing. Let's take, okay, this is a hen. Okay, this is a hen. Okay, this is a hen. This, let's say, lays around 20 eggs in a month. Okay, and this is one more hen. Okay, which lays 10 eggs. But this hen is good, like hefty, and this hen is thin. Like, what I'm trying to say is, if a fried chicken company is looking for hen, obviously they will choose this. But I am a farmer and I have I want to make something best out of it. So what I'm going to do, I'm going to take the genes from this that helps this hen to lay 20 eggs, and I'm going to take the genes from this that makes the hen hefty, and I'm going to do a cross and I'm going to get a hen that is hefty at the same time going to lay 20 eggs. Right. So this way, even in plants also these people do it, right? Even in plants also these people do it, and that is called as artificial hybridization, where they form superior varieties. Clear or not? Where they form superior varieties. Yes? Clear? Is the voice getting cut, students? From this, that makes the hen hefty, and I'm going to do a cross, and I'm going to get a hen that is hefty. The same time. Yeah, okay. It's clear only. Okay, so have you guys seen such a beautiful hen ever in your life? If yes, please put it in the chat box, okay? This is the most precious hen that I've ever drawn. Okay, so I'll write it here. This is hen. Now look here. To combine desirable characters to produce commercially superior variety. So now I have made a superior variety where it is hefty plus who is going to lay 20 eggs. Now same thing is going to happen in plants, but how are they going to do it? Look here. Artificial hybridization is one of the major approaches of crop improvement program. Okay, so in such crossing experiment, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination. So what they are going to do, okay, what they are going to do, pollination, if pollination has to happen, I will do one thing. I'm going to make sure that the pollen grains that are going to land on the stigma is in such a way that they are not going to be, like some random pollen grain. They are going to be of superior quality. So I am going to get a better yield. Okay, I am going to get a better yield. Understood? So this is achieved by emasculation and bagging techniques. Emasculation and bagging means what? I want to make sure that unnecessary pollen grain does not fall. Okay, or I want to make sure that this pollen grain does not do self-pollination as well. I will remove the anther, right? I will dust out the pollen grains. I can do different things. So look here. If the female parent bears bisexual flowers, removal of anther from the flower bud before the anther dehiscent using a pair of forceps is necessary. This step refers to as emasculation. So emasculation is what, students? Removal of anther. From what flower? A bisexual flower, right? From a bisexual flower. That is, if this flower has both anther and stigma, and there is high chances that this people will do, self-pollination, then we have to do one thing, that is to remove this anther, remove the anther before this dehiscence happen, because the pollen grains will land on the stigma. Is this clear? Okay, chalo. Now that is called emasculation. Now the emasculated flower, have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. Yes, ma'am, you have done something to remove self-pollination, or you have taken a step to do, get rid of self-pollination. But what if by wind, water, insect, humans, or lizards, garden lizards, what if the pollen grains are going to get scattered, which are unwanted? So that time we have to protect this. So what we are going to do, when the stigma is there, I remove the anther. So let's just erase off the anther. Okay, let's erase off the anther. Once I remove the anther, I have to make sure that the stigma is bagged. Okay, and what paper I'm going to use? Butter paper. Okay, I'm going to use a butter paper, and this is called as bagging. So emasculation is followed by bagging, then cross pollination will happen, again rebagging is going to happen. Okay, so look here, this process is called bagging. When the stigma of the bagged flower attains receptivity, that is, when stigma is ready to take pollen grains, we will be like, okay, we will dust some pollen grains. Okay, so mature pollen grains collected from the anther of the male parent are dusted on the stigma, and the flowers are rebagged. Is this clear to all of you? Very baby thing. Okay, Roshni, you joined late, so you can make a note of this. Okay, this is double fertilization, syngamy plus triple fusion. Those who joined late, please make a note of this. Okay, later don't complain telling ma'am you did not send the notes. This is very, very important. Okay, yes. Dikshi is showing me, ma'am, this is how hen looks, not like this. Okay, this is a hybrid of hen plus man. Okay, so do not disrespect students. I only made the hybrid. Okay. Notes you will not get Dikshi because this is handwritten. This is written with a pen. No, I cannot share it. When I share it, it will look all small, small. It will look. Okay. Yes. My own vocabulary sometimes comes. None. Okay. Now here different shapes of embryo are there. Let's see that also. So students, see, once the fusion has happened, okay, the synergids are going to degenerate. Okay. Zygote will be present. Okay, primary endosperm cell, that is where the central cell was there. No, instead of that, primary endosperm cell will be there, PEC will be there, then pen will be there, primary endosperm nucleus, then degenerating antipodal cells. You can just imagine, no, degenerating antipodal cells. Then students, see, the zygote is going to continue the division, okay, and continue the division, and it will form. Don't you think I have everything customized? I have my own series called as GM, what was it? GMERT. I think I'm right. No, this is what he tells. I have my own dictionary. Okay, GM dictionary. I have my own art, GM art. So I am basically a company by itself. Everything I only create. Students, in my class, you will see a lot of hybrid animals which I only made. Yes or no? Tanuja, Roshni and Dikshi, do you agree? Do you guys agree to this? Okay, so students, once the fusion has happened, pect is going to be there, pen is going to be there, zygote is going to be there, and zygote will undergo different stages of growth. Okay, different stages of growth, and that stages will be, you can have a globular embryo, you can have a heart-shaped embryo, plumule, and radicle. That is, shoot and root has to be formed. No? So they'll be getting ready for that, and upon the same time, cotyledons also will be there. Okay, agreed, ma'am. Okay. Post-fertilization structures and events. So during all this while we have learned pre-fertilization. Tell me, what and all did we learn in pre-fertilization? In pre-fertilization, GM production. Okay, pre-fertilization we learned about how we can form the gametes. Yes or no? How we can form the, I'll write it this side, no? Okay, students, during pre-fertilization, we learned about forming gametes. That is, we learned about microsporogenesis, and megasporogenesis. Okay, megasporogenesis. Here we form the male gamete, that is up to pollen grain, male gametophyte. Okay, in that we form the generative cell or the male gamete. In megasporogenesis we form the embryo sac. Okay, inside the embryo sac egg was formed. Correct? Yes, very good. Microsporogenesis, megasporogenesis. Now the next event that we studied was fertilization. Fertilization, what all did we study? We learned about double fertilization. Okay, double fertilization. We learned in between only, we learned outbreeding devices. I'm just writing the main topic, okay, what we learned. So you remember outbreeding devices. Yes, done. Now we are going to the post-fertilization event. That means everything has happened. Now we have to see what they are going to form. What is the ultimate result that is going to come out of it? Okay, what is the ultimate result? Look here, post-fertilization events. So following double fertilization events, the events of endosperm embryo development, maturation of ovules into seed, ovary into fruit are collectively termed as post-fertilization event. Now let's see what is going to happen to the endosperm. Endosperm development precedes embryo development. Okay, why? The primary endosperm cell divides repeatedly and forms a triploid. Okay, they will continuously divide and form a triploid endosperm tissue. The cells of the tissue are filled with reserve food materials and are used, okay, and are used for nutrition of the developing embryo. So as I already told you, that everyone needs something that will give them the, what to tell, the nutrition. So here it is going to be the PEN, okay, primary endosperm nucleus. So what they are going to do, students, they will have reserve food materials, okay, reserve food materials plus nutrition they are going to get. Now in most common type of endosperm development, the pen undergoes successive nuclear division to give rise to free nuclei. Okay, so here what they are talking about, always remember endosperm can be of 3 types. Okay, endosperm can be of 3 types. It could be nuclear, it could be cellular, yes, okay, lobia. Now this you can completely skip, this term is never coming in your chapter, but these 2 terms somewhere you can see. Okay, now nuclear means what, students? The division happens and there is a free nuclei. Okay, so this stage of endosperm is called as free nuclear endosperm. That means what? The pen will undergo nuclear division. Is this clear to all of you? Pen will undergo nuclear division. Okay, now cellular means what? The primary endosperm will be followed by cytokinesis. That is, the cytoplasm is going to, the cytokinesis is going to happen. Okay, and therefore the endosperm becomes cellular from the beginning. Example are coconut. Coconut is the example. Palm is an example. Here, nuclear endosperm, what is going to happen? Okay, successive nuclear divisions are going to happen. Okay, successive nuclear divisions are going to happen. I'll draw a diagram and show you best explanation possible. After that, you will not have any doubt. Okay. Now let's see where I can draw the diagram. I think I've left an empty page somewhere, students. Here I'll show you. Okay, now here nuclear division. This question will come, that's why I'm drawing it. Nuclear division means, let's take, this is the cell. Okay, very beautiful cell. Okay, this will happen. So nuclear division means free nuclear divisions are going to happen, right? Successive nuclear divisions are going to happen. Very good. So like this, nucleus will keep dividing. Right here. Okay. Like this, nuclear division is going to happen. Can you think of it? Right? Like this, nuclear division is going to happen. I'm not drawing the numbers correctly. Okay, but you should know that the numbers. So like that. Like this, nuclear division will happen. So this will give rise to free nuclear division, like in wheat and all. Okay, wheat, you can think of the example. Okay. Yes, all of you? Yes. Wheat, rice, maize, all of these are examples. Now at the same time, what is cellular, ma'am? Cellular is going to be, yes, can you see here? Again, this is the pen, okay? We are only talking about endosperm, don't get confused with zygote. Okay, zygote we haven't spoken. Yes, very good, okay? Okay, like this, endosperm, okay, cellular division is going to happen. Then, I mean, here not cellular division, yeah, cellular division like this. Okay, division will continuously happen, and this is going to happen. Okay, my birthday. It's in the, it's not coming anytime soon. Okay, again it will come only on next year. It is over. There's a video on our channel, by the way, on my birthday. This is clear. Okay, now This one, actually we shouldn't draw this, we don't have to draw this, they're just cellular divisions. Okay, so students, is this clear? Cellular divisions, all the cells are going to separate like this. Okay, this is a very diagrammatic representation to make you understand. Now let's go further. Let's see. Okay, free nuclear endosome. Subsequently, the cell wall formation occurs and the endosperm becomes cellular. Understood? So, till that last stage, you saw that endosperm was there. Okay, endosperm was there. Then slowly cellular division started coming. Okay, the number of free nuclei formed before cellularization varies greatly. The coconut water from the tender coconut that you are familiar with is nothing but free nuclear endosperm. Okay, free nuclear endosperm made up of thousands of nuclei, and surrounding white kernels is your cellular endosperm. So in coconut you can see both nuclear as well as cellular divisions. Okay, so endosperm may be neither completely consumed by the developing embryo like in your pea, groundnut and beans before the seed maturation, or it may be persistent in a mature seed, that is castor and coconut, and it is used by seed germination. Okay, so here they are trying to say it can be endospermic, like where entire endosperm is consumed, ex-albuminous or albuminous seed. Okay, now students, embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygote divides only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrients. That is, I do not have Tanuja, one more day less, more. Okay, 16th. Okay, so what is going to happen, students? Zygote division will happen only once they have got confirmation that okay, there is amount of endosperm. By simple thing, before you go to some place or a hotel, you check for hotels, restaurants near that place, so that you do not go hungry, you have enough food to survive. Same way here also, before the zygote is going through any kind of changes, they will check if the endosperm is formed. Got it? Now, this is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly, the early stages of embryo development, that is embryogeny, are similar in both monocots and dicots. Now, depicts the stages of embryony, a dicotyledonous embryo. The zygote give rise to pro-embryos and subsequently to globular, heart-shaped and mature embryo. Okay, there is going to be 2-3 stages. That is nothing but your pro-embryo stage. You can all look at the diagram in your textbook. Pro-embryo stage. After that, maybe a globular, then a heart-shaped. Okay, then a heart-shaped, then mature. Got it? Okay, fine. Now, a typical dicotyledonous embryo consists of an embryonal sac and 2 cotyledons. Okay, so that is going to be like this, students. What they're trying to talk is, students, how do you find my botany class? Because you already know about zoology classes. See, this is the cotyledon, that these are 2 cotyledons. Okay, and this will give rise, this is the plumule. This will give rise to the shoot. This is going to be the radicle that is going to give rise to the root. Okay, right? So here they're talking about, this is the embryonic axis. Middle embryonic axis, cotyledon. Okay, the portion of the embryonal axis above the cotyledon is called as your epicotyle or the plumule, right, which terminates into plumule. So epicotyle, and here it is going to be hypocotyle. Okay, hypocotyle. Okay, yes, okay, nice, you people like it. So 2 cotyledons are there, very simple. 2 cotyledons are there. Between that, one embryonal axis is coming. Whatever is above the cotyledon, we will call it epicotyle, which will terminate in the plumule. That plumule will give rise to a shoot. Whatever is below the cotyledon, it is called as hypocotyle, which will terminate in a radicle that will give rise to the shoot. Okay, Understood? Now, the portion of the embryonal axis above the level is understood plumule epicotyle. Now, the cylindrical portion below the cotyledon is called as hypercotyle, which will terminate in a radicle or a root tip. Okay, but remember that the root tip has a root cap. Why they have a root cap? Simple thing, because the root has to go through hard soils, go through surfaces where there is no water. If you would have seen, some plants even grow in concrete road. Yes or no? You will see that some plants are growing between the concrete road. That is because this root cap is very strong. It will go through all the tough, like all the hard substance, it will go without damaging the root. Okay, chalo. Yes, clear? Okay, now embryos of monocotyledons possesses only one cotyledon. Yeah, what does monocot mean? Mono means one, cot means one cotyledon. In the grass family, the cotyledon is called as scutellum, that is situated towards one side of the embryonal axis. At the lower end, the embryonal axis has a radial root cap enclosed in an undifferentiated sheath called coleorhiza. Okay, I'll try to look and draw the diagram, students, because the diagram is little complicated. Okay. There's two diagrams. One is given in your textbook, but that is not clear. So I will just draw one more diagram, okay, which is going to be a little hard to draw without seeing. You don't have to draw, okay. First of all, you don't have to draw. So usually we do not draw this diagram, we draw the easy diagram, but since I did not add the diagram, okay, it looks something like that. I did not add the diagram, I thought I'll draw this. Okay, students, now in between so many things are there which I definitely cannot draw. Okay, but okay, like this, all of you can look at the structure in your textbook, right? Something like this will be there. Okay, now this is not the important part. The important part is to okay, now this portion, right? This portion is the scutellum. This portion is the scutellum. Now, since this portion is the scutellum, this portion, right, which is going to give rise to the plumule, there is a protective layer called a coleoptile. And this portion that will give rise to the root, there is a protection called as coleorhiza. Okay, coleorhiza. Now, below this, there is going to be shoot apex, root apex, and epiblast, and all of that is going to be there. But what you have to understand is this portion, okay? This portion, that there is scutellum, which will actually give a differentiation between your coleoptile and coleorhiza, or a differentiation between your plumule and a root. Okay, so here you can see at the lower end, the embryonal axis, there is going to be undifferentiated sheath called coleorhiza. So here, let's say, students, this is going to be the root cap. Okay, this is going to be the radicle, etc. So, the portion of the embryonal axis above the level of the attachment of scutellum is called as epicotyle. This is the epicotyle region. Okay, epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow follicular structure called coleoptile. You don't have to worry about all of this. Just remember only this much. Coleoptile protects the shoot apex. Okay, protects the shoot apex or the plumule, and this plumule gives rise to the shoot. Simple, yes or no? Then coleorhiza is the portion that will protect the root or the radicle. Okay, and this will give rise to the root. Clear? This much you remember. Then you remember about scutellum. Scutellum is the portion that gives that differentiation. Done, all of you? Yes? All of you tell me. Yes. Okay. I've added another diagram. I'll show you that. Now we'll go to seed. Students, before that we'll take a slight break. Okay. I'll just okay, I'll just have something in come. Okay. We have seed, we have pericard, dormancy. Okay, very few portions again, fruits, that's it. Okay, another maybe 30 minutes portion is left. So I'll just have breakfast income. I can, all of you also can go have breakfast income. Okay, 20 minutes break. Fine. Hello students, am I audible? Hello students, am I audible? Yes, audible, right? Okay, so we will continue with seed. So we understood endosperm, we understood two types of endosperm is there, nuclear and cellular. Now we're going to understand seed, because the whole point of doing this entire sexual reproduction is why, so that we get the seed and the fruit right out of it. So here in angiosperm, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. So seed is formed from the ovule. This all of you know, no? A fertilized ovule give rise to seed. Simple as that. Okay. Now, seeds are formed inside the fruits. A seed typically consists of a seed coat, cotyledon, and an embryo sac. Okay, embryo sac. Now the cotyledon of the embryo are simple structures, generally thick and swollen due to the storage of food reserves. Mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm. So before also they had spoken about this, telling that seeds can be of two types. Okay, non-albuminous. Yes, correct, Roshni. Non-albuminous and ex-albuminous. I mean, sorry, non-albuminous and albuminous. Okay. So here, non-albuminous seeds means they have no endosperm left. That means the embryo has consumed all the endosperm. So they have no residual endosperm. Okay, they have no residual endosperm. That means they do not have any more endosperm. All the endosperm has been used up by the growing embryo. Okay, and it is completely consumed. Okay, no residual endosperm. It's completely consumed during the embryo development. Example, you can remember pea plant, groundnut. Okay, example, pea plant, groundnut. Now, albuminous seed retain a part of the endosperm as it is not completely used. So here what is going to happen? The endosperm will not be completely used. Endosperm not completely used. Okay, some will be remaining. Okay, got it? This is example is wheat, maize, barley, etc, etc. Now occasionally in some seeds such as black pepper, this is very important. Okay, BB, that is black pepper and beet. Okay, black pepper and beet, what is going to happen, students? Perisperm you'll see. That means a persistent nucellus. Now where did nucellus come from? All of you remember, in the embryo sac nucellus was there. Okay, that was providing the nourishment for the growing embryo sac. So when there is persistent nucellus, we call it, okay, persistent nucellus, we call it perisperm. This question has come many times in NEET PYQ, so remember this, it's a PYQ question. Okay, it's a PYQ question. Now, integument. So now what is happening is the embryo sac structure you have learned, right? Now they are talking about every single layer of the embryo. What did it end up? What is the fate of all of these people? Okay, so look here, integuments of ovules harden as protective seed coats. So they're telling the integuments that we saw in our anatropous ovule. Okay, integuments, what has they become? They became seed coat. They became seed coat. And those seed coats, see, the micropyle remains as a small pore. So what is the fate of the micropyle? Micropyle had an opening where the integuments were not present, so micropyle still remains as a small pore in the seed coat. Okay, the seed coat. This facilitates the entry of oxygen. So micropyle is going to help in the transport of oxygen and water into the seed. That means for that exchange, no small pore is left. Now, as the seed matures, as the seed matures, its water content is reduced and the seeds become relatively dry, 10 to 15% moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state inactivity called as dormancy. That is, this embryo, students, can become into a dormant state. That is like how we saw it in animals, hibernation and aestivation. Like that itself, the embryo also can go to a dormant stage. Okay, it can go to a dormancy stage when the conditions are unfavorable. That is a stage where the seed goes to sleep. No, like that we tell the seed will be completely inactive. Okay, it will not do any germination or anything. It will go to a complete inactive stage until the favorable condition happens. The seed will remain in that stage. Okay, I hope this is clear, all of you. Okay, now here, okay, if the favorable conditions are available, adequate moisture, oxygen, suitable temperature, they will germinate. So from this stage, if they have to come back to a germination stage, then all the condition has to be good. Okay, germination stage, all the condition has to be good. Now look here, as ovules mature into seeds, the ovary develops into fruit. That is, the transformation of ovules into seed and ovary into fruit proceeds simultaneously. The wall of the ovary develop into the wall of the fruit, which is called the pericarp. So the integuments have become the seed coat. Okay, and the fruit has become, the fruit wall is made up of pericarp. So the fruit, the wall of the ovary, remember like this is the ovary. Okay, inside that ovule was there, right? Inside the ovule. So the wall of the ovary became pericarp. Okay, wall of the ovary became pericarp. Now, the fruits may be fleshy as guava, orange, mango, etc., or may be dry as groundnut and mustard. Many fruits have evolved the mechanism of dispersal of seed and recall the classification. Okay, you can remember the classification. Any doubts so far, students? Any doubt, please ask me right now. Any doubts? Okay, I'm also okay, clear? Okay, fine. Yes, clear, fine. Okay, now if you see, it's not mentioned in NCERT, but in case you want to know, the seed coat is made up of testa and tegmina. Tegmen, okay, not tegmina. Tegmina is your forewing. Okay, tegmen, testa is the outer, tegmen is the inner. Okay, got it, all of you? Yeah, okay. Chalo. Now the outer layer will be a little more thick to protect the seed, inner layer will be a little more thin. Okay, now types of fruits. Okay, types of fruits. So in most plants, by the time the fruits develop from the ovary, other floral parts degenerate and fall off. However, in few species such as apple, strawberry, cashew, the thalamus also contributes to the fruit formation. That is, see students, very simple definition. If the fruit is formed from the ovary, what is the fate of the ovary? Fate of the ovary is like you have to become the fruit. What is your fate? To become a doctor. Now, if you're not becoming a doctor, instead of becoming someone else, will you be called a doctor? No. You will be called according to your profession. Now, if you become a teacher, you'll be called a teacher. If you become an educator, you'll be called an educator. If you become a lecturer, you'll be called a lecturer. Now, if I'm going for a doctor job, my fate is like, okay, I have to be a doctor. If I don't become that, then I'll not be called a doctor. No. So same way, ovary should become fruit. Anything other than the ovary, okay, any other part, any other part other than the ovary, okay, other than ovary will be forming a fruit, okay, will form a fruit, then it will be called as false fruit. Okay, false fruit. Understood? So if thalamus is becoming a fruit, or if the receptacle is the same thing, thalamus receptacle is becoming the fruit, or something else is becoming the fruit, then this will not be called as a true fruit. So such fruits are called false fruit. Most fruits, however, develop from the ovary and they are called as true fruits. Okay, although in most of the species fruits are a result of fertilization, there are few species where fertilization does not happen. Okay, so in this diagram also the structure is given. You can see the cotyledon. Okay, here you can see the hypocotyle, endosperm, seed coat. Okay, this you can, this and this too you can focus. Coleoptile, coleorhiza, radicle, plumule, endosperm, scutellum and pericarp. You can see, you know, scutellum is giving a differentiation between the endosperm and your embryo. Endosperm and embryo, that difference is given by your scutellum. Okay, difference is given by the scutellum. Then you can see how the thalamus is there, seed is there, okay, and the endocarp is the false fruit. Now look here. Some of the fruits develop without fertilization, and such fruits are called as parthenocarpic fruits. What are they called? Parthenocarpic fruits, so which develop without fertilization. Banana is one such example. Parthenocarpy can be induced through the application of growth hormones. Such fruits are seedless. Okay, seedless, nowadays seedless watermelon is there, seedless papaya is there. All this is done for us, our ease, for our ease, and that is done with the help of parthenocarpy. If fertilization does not happen, and seed formation will not happen. So like that we can avoid the seeds as well. Seeds often several advantages to the angiosperm. Firstly, since the reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategy for dispersal to new habitat and helps the species to colonize in other areas. Okay, now here they're talking about some, you know, common factors, but there are some names which are given to you which they might ask in questions. Okay, look here. As they have sufficient food reserves, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides the protection for the young embryo. Being products of sexual reproduction, they generate new genetic combinations. Simple as that, variation. Okay, variation is possible. Now, seed is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for the storage of seed, which can be used for food throughout the years. Nowadays, all of you know, seed banks are there. Okay, government itself has seed bank, private seed banks are there, where they'll give a packet of seed to the farmers and the farmer will go and cultivate the seed, okay, throughout the year and also raise the crop in the next season. Can you imagine an agriculture in the absence of seed? It's going to be definitely difficult, because now banana now is grown by vegetative propagation. Now we cannot grow everything by vegetative propagation. There are now rows, we do grafting and all of that, but there are a lot of plants which are grown by seeds. Okay, how long do the seeds remain alive after they are dispersed? This period again varies greatly. In some few species, the seed loses viability within a few months. Like we saw in Rosaceae, Leguminosae, Solanaceae, up to few months the viability will be there. But at the same time, if you see rice and wheat, they have only 30 minutes of viability. Okay. Seeds of a large number of species live for several years. Some seeds can remain alive even for hundreds of years. There are several records of very old yet viable seeds. The oldest is that lupine. Okay, that is Lupinus articus, evacuated from the Arctic tundra. The seed germinated and flowered after the estimated record of 10,000 years of dormancy. Can you imagine 10,000 years of dormancy? A recent record of 2,000 years old viable seed is of the date palm, Phoenix datilifera, discovered during the archaeological excavation at the King Herod's palace near the Dead Sea. Okay, this just look at the names. You don't have to by heart or anything. Just look at the names. Okay. Now we're coming to the last topic of this chapter, students, apomixis and polyembryony. Although seeds in general are products of fertilization, a few flowering plants, such as some species of Asteraceae and grasses, have evolved a special mechanism to produce seeds without fertilization. Okay, so when, see, understand the difference. Fruits without fertilization is called what? Answer me, all of you. Fruits without fertilization is called what? Which line, Akhtar? Which line? Fruits without fertilization is called what, students? Parthenocarpy. Okay, parthenocarpy. Parthenocarpic fruit. Now, students get confused here. Okay, seed without. Okay, seed. Very good. Very good. Who is that? Tanuja and Roshni. Very good. Seed without fertilization is called what? Students get confused here, and let me tell you, it's a very common thing to get confused. There is no mistake in getting confused, but you have to have this clarity. Seed without fertilization, students? Seed without fertilization? Yes, very good. Seed without fertilization. The answer is on the board, on the screen. Seeds without fertilization. Seeds without fertilization. Very good, very good. Apomixis. Apomixis. This difference, if you understand, you will never get confused. Fruit without fertilization. Parthenocarpic fruit. Example, banana. Seed without fertilization. Apomixis. Where is its seed? This example is going to be grasses. Example, grasses. Here, example, banana. Got it, all of you? This difference, if you understand, more than enough. More than enough. Now, what is fruit production without fertilization called as? I already asked you. Apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of an apomixic seed. In some species, the diploid egg cell is formed without reduction division and develop into embryo without fertilization. More often in many citrus and mango varieties, some of the nuclear cells surrounding the embryo sac starts dividing, protrude into embryo sac and develop into embryo. So what they are trying to say, nuclear cells are there, no students? Okay, nuclear cells, they will undergo division and they will start surrounding. Okay, your nucleus, no, like that, they will surround the embryo sac. Surround the embryo sac. And here they will start dividing and they will protrude into the embryo sac and develop the embryos. Like waiting, instead of waiting for fertilization, what they will do, they will only become the cells. Okay, they will only become the cells and those cells only will form embryo. In such species, each ovule contains many embryos. Right now, if you have seen in orange, you know, many embryos are there, no? So in such situations, what happens? Many embryos will form and that is called as polyembryony. Polyembryony. Got it? When many embryos are formed. Okay, when many embryos are formed. Got it, all of you? Example is citrus and mango. Okay, citrus and mango. Chalo, yes ma'am, clear? Here they have given some pictures, so I was just seeing that. Okay, some pictures they have given. Okay, now this is done. Hybrid varieties of several food and vegetable crops are being extensively cultivated. Cultivation of hybrids have tremendously increased the productivity. One of the problems of hybrid is that hybrid seeds have been produced every year. If the seeds collected from hybrids are sown, the plants in the progeny will segregate and do not maintain hybrid characters. So production of hybrid seeds, okay, production of hybrid seeds is costly and hence the cost of hybrid seeds become too expensive to farmers. If these hybrids are made into apomix, there is no segregation of characters in hybrid progeny, then the farmers can keep on using hybrid seeds to raise new crops every year and he does not have to buy hybrid seeds every year. Because of the importance of apomixes in hybrid seed industry, active research is going on. That is, many people, okay, many people do apomixes in an to make it very convenient. Okay, to make it very convenient. Now students, if I draw a fruit here, you just tell me which are these layers. Wow. Okay. Yes. Students, see, first layer is going to be epicarp. Yes or no? Epicarp. Okay. Then what is going to be there? Outer layer, we can also call it as exocarp. Okay, exocarp. This, see, exocarp. This layer, this outermost, this inside layer, this coat is going to be called as pericarp. Okay, pericarp. Then this fleshy area is going to be mesocarp. And the seed, right? The seed will be there. Seed. Yes, very good. Exocarp, epicarp. Okay, you can also call it as epicarp, no problem. Exocarp is the skin, mesocarp is the fleshy area. Okay, the fleshy portion, and then your endocarp is going to be an inside portion where the seed is there. Okay, like this, different fruits is going to be different, so try to understand that. You will learn this in morphology, that's why I'm not going to teach you in depth. Okay, teach you in depth. So students, we have completed the chapter, all the portions I've done. Okay, so just understand the difference. I'm just going to this portion. Okay, I'll just go through the summary, give me 2 minutes. Okay, just here only these things you remember. True fruits. Okay, true fruits, it's formed from the ovary. Formed from ovary. Okay, false fruit, formed from any part other than ovary. Okay, formed from other parts except ovary. Except ovary. Got it? Except ovary. Then you have parthenocarpic fruit. Parthenocarpic fruit, that is basically fruit formed without fertilization. Fruit without fertilization. Okay, then we have apomixis. Thank you. Apomixis, that is seed without fertilization. Seed without fertilization. Then we have polyembryony. Okay, polyembryony, that is many embryos. Yes, very good, very good. Okay, all of you are answering. Many embryos are formed, and these embryos are formed from the nuclear division. Okay, not from the fertilization, from nuclear division. That's why many embryos have been formed. Okay, yes or no? Is it clear? So here true fruits are there. If you want to write one examples, you can go for that. You can write example true fruits. See, the most of them are true fruits, right? The false fruits are very less of them. Yes or no? And students, pericarp, I told you about pericarp, no? But in case you have doubt about who, like in the sense, like in morphology, you will see difference between mango and coconut. It's a drupe fruit. Okay, so like that. Now here false fruit, you can write apple, strawberry, cashew nut. Okay, parthenocarpic fruit, banana, and any of the seedless fruits. Okay, apomixis, you can write the grasses or the Asteraceae families. Polyembryony, you can write citrus fruits. Okay, citrus. Clear? One example also I have asked. Yes or no? Clear? Chalo. We'll go quickly through the summary. Just give me like 5 minutes. This will do a revision. Okay, this will do a revision. See, flowers are seat of sexual reproduction in angiosperm. In the flower, angiosperm consists of stamens, which represents the male reproductive organ. Okay, stamen has anther and filament. Gynoecium consists of pistil that represent the female reproductive organ. A typical anther is bilobe, dithecous, and tetrasporangiate. Students, after studying everything, try to read summary, because summary will give you like a short note. Okay, see, they'll directly have given bilobe, dithecous, tetrasporangiate, so it's a good thing. Okay, now pollen grains develop inside the microsporangia. 4 wall layers, that is the epidermis, endothelium, middle layer, and tapetum, surrounds the microsporangium. Cells of the sporogenous tissue lying in the center of the microsporangium undergoes meiosis to form the tetrads of microspore, or you can write MMC, that is microspore mother cell undergoes meiosis and forms the microspore tetrad. This microspore tetrads together with the help of the callus, which will be removed with the help of callase that is given out by the tapetum. Individual microspores mature into pollen grains. Pollen grains represents the male gametophytic generation. The pollen grains have 2 layered wall, the outer exine and the inner intine. The outer exine is made up of sporopollenin, which is one of the most resistant organic matter found, which cannot be dissociated or which cannot be damaged by acid, alkali, or temperatures. And they have germ posts. There are places where sporopollenin is absent, so that the pollen tube can germinate from there. Okay, and then we have intine, which is the innermost layer, which is made up of cellulose and pectin. Okay, now the pollen grains have 2 cells, one vegetative cell, one generative cell. The vegetative cell will give all the food reserve for the generative cell. Okay, the generative cell will have 1 male gamete, or they can have 2 male gametes, depending on if it is a 2 cell stage or a 3 cell stage. So look here, 2 cells, a vegetative cell and a generative cell. 3 cell stage, a vegetative cell, 2 male gametes at the time of shedding. Is this clear? Now, the pistil has 3 parts, the stigma, style, and ovary. Ovules are present in the ovary. The ovules have a stalk called as the funicle. Protective integuments are there, and these integuments later become the seed hard coating, okay, around the seed. Now, they have an opening called as micropyle. Micropyle becomes a small pore in the seed, which allows for oxygen and water exchange. Now, the central tissue is called the nucellus, which provides the development or provides the nourishment for the growing embryo sac. If the nucellus is persistent, that will be called as a perisperm, which is seen in beetroot and black pepper. Okay, in which the archisporium differentiates. A cell of the archisporium, that is the megaspore mother cell, divides meiotically, and one of the megaspore forms the embryo sac. So out of 4 megaspore mother cell, only one of them becomes, yes, exactly, GM express. Out of 4 megaspore mother cell, only one become functional, the rest 3 will degenerate. That one functional will be forming the embryo sac, which will have the eggs and the synergid, which is a 7 cell 8 nucleate stage. At the micropylar end, the egg apparatus will consist of 2 synergids and an egg cell. Okay, they will have 2 synergids and an egg cell. At the chalazel end, they will have 3 antipodal cells. So opposite to the micropylar end is the chalazel end. At the center is a large central nuclei with 2 polar nuclei. Pollination is a mechanism of transfer of pollen grains from anther to stigma of the same flower or from a different flower. So depending on that, it will be called as autogamy, geitonogamy, or xenogamy. Now, autogamy is where self-pollination happens. Geitonogamy is between in the same plant, 2 different flowers are going to pollinate, and then you have xenogamy, which is cross pollination by itself. Okay, clistogamous, chasmogamous flower, try to remember. Now, pollination can happen with biotic or abiotic factors. Biotic factors are insects mainly, and then comes animals, lizards, etc. That is called as entomophily. If it is by water and wind, then they have different name, anemophily and hydrophily. Wind is the most common kind of pollinating agent. Okay. Okay. Students, someone wants to do photosynthesis in the chat box. Please respect that person. Okay, his name is Nala Natik. Okay. Pollen pistil interaction involves all the events from landing of the pollen grains on the stigma until the pollen tube enters the embryo sac. Such uneducated people are there in the world that the moment they see sexual reproduction in flowering plants, the moment they see the word sexual reproduction, they think they have only one concept about it. I don't know how these people are even given birth to. Okay, so I think his mother should be very proud. Okay, such common senseless creatures. Flowering plants he cannot see. Okay. Okay, so look here, students. enters the embryo sac when the pollen is compatible, or the pollen inhibition when the pollen is incompatible. See, following, so pollen pistil interaction happens depending on the compatibility, that is the pollen grains, if they are compatible. Okay, if they are compatible, they are going to continue the pollen tube formation. If they are non-compatible, they will get rejected. Okay, they will get rejected. Okay, they will get rejected, and those rejection is because that they are of the wrong type. Okay, just like this guy, wrong type, rejected piece. So like that, like rejection will happen. The right type will continue to develop, continue to have a good future. The wrong types will get rejected by the earth itself. Same way, even the pollen pistil interaction happens, the right one will be allowed to germinate and form the pollen tube. And one second. Okay, now students, what is going to happen when the pollen is so following the compatible pollination, the pollen grains germinate on the stigma, resulting in pollen tube growth through the style, and they enter into the ovule. Finally discharges two male gametes. Okay, two male gametes in one of the synergids. So very simple, students, both the male gametes, if it is in a 2-cell stage, only one male gamete will go and fuse with the egg. But if it is in a 3-cell stage, both the male gametes will be put into the synergids, and with the help of filiform apparatus, the pollen tube will go inside, and the male gamete, one male gamete will fuse with the egg, and another male gamete will fuse with the polar nuclei. The one that fuses with the male gamete is called as the zygote, and the one that fuses with the polar nuclei will be called as PEN, that is primary endosperm nucleus. Primary endosperm nucleus further divides, becomes the endosperm, and the zygote will further become embryo. The endosperm is going to give all the nourishment for the growing embryo. Clear? Chalo. Now, angiosperm exhibit double fertilization because two fusions event occurs in each embryo sac, namely syngamy and triple fusion. Syngamy means it is between the egg and the male gamete. Okay, the products of these fusion are diploid zygote and the triploid primary endosperm nucleus. Okay, zygote develops into embryo, and the primary endosperm cells forms the endosperm tissues. Formation of endosperm always precedes the development. Okay, always precedes the development. Now, the developing embryo passes through different stages. Okay, the developing embryo passes through different stages such as pro-embryo, globular, heart-shaped stages before the maturation. Mature dicotyledon embryo has two cotyledons and an embryonal axis, which is called the epicotyle and the hypocotyle. Clear? Epicotyle and the hypocotyle. Chalo, clear all of you? 1 second, students. Okay. Now, embryos of the monocots have single cotyledon. After fertilization, the ovary becomes fruit and the ovule becomes seed. That is the general thing. Okay, ovary has to become fruit. If ovary does not form into fruit, or some other part of the plant is becoming a fruit, like the thalamus, then we will call it false fruit. If it is forming from the ovary, we will call it the true fruit. Okay, and the ovules will become seeds. Now, the fate of integuments become the hard seed coat, and the micropylar end becomes the small opening on the seed that allows oxygen and water exchange. Okay, then what else, students? Did we miss out on something? The fruit wall, the ovary wall will become the fruit wall, which is called the pericarp. Perfect. Now, embryos of the monocot has single cotyledon. After fertilization, ovary develops into fruit, ovule becomes seed. A phenomena called apomixis is found in some angiosperm, particularly in grasses. It results in the formation of seed without fertilization. Apomix have several advantages in horticulture and agriculture. Please read that paragraph once again. I did not go through it thoroughly. Okay. No, it will start at 4, Tanuja. Mostly at 4, our max up to 4:30. Okay, but it will start because I have to finish this class, then I have to travel to office, so that timing might change a little. Okay, so just inform others also when they join that in case this thing is shifted, it will be shifted by half an hour. That's all. Okay, 4:30 itself I'll start. Okay, because I have to travel, and if there is a lot of traffic, then it will take me more time to travel. Okay, so some angiosperms produces more than one embryo, and to this seed, this phenomena is called polyembryony, which we see in citrus plants. Okay, which we see in citrus plants. So that is, many embryos are formed. Now, how these embryos are formed? By nuclear division. Nuclear division will happen. They will protrude into the embryo sac, and they will start forming embryo. Because of it, multiple embryos will be formed. Clear, all of you? Any doubts so far? So students, sexual reproduction is done. This definition, please try to remember. These are the major definitions. At the same time, double fertilization. I will also check Tattva if you have anything. And you guys want to answer one or two questions from Tattva? Any of you? You can just put the short form when I'm asking question. Please, I can't repeat. Okay, please, I can't repeat. Yeah. So students, first question to all of you. Okay, dash cells of the pollen grain divide to form two male gametes. Two male gametes. What happened, Tanuja? Yes, students, tell me. Dash cells of the pollen grain will form two male gametes. Tell me quickly. Please answer, students. Please. Where do you guys come? I think the chats are slow. Generative cell. Roshni, one answer. Generative cell. Vegetative cell is providing nourishment. It is always going to be the generative cell. Okay? It's always going to be the generative cell. So next question. All zoo creatures are waking up now and coming on the chat box. So in angiosperm and ovule represent what? Very good question. In angiosperm and ovule represent what? Options are megasporophyll, megasporangium, a megaspore mother cell. And in an angiosperm, okay, an ovule represents dash. Good question. Yes. Okay, I think the chats are slow, students. I cannot do the questions then. Okay, I cannot do the questions because the chats are very slow. It will take too much time. Megaspore. Okay, megaspore. Because megaspore, that megaspore only is becoming the ovule, the embryo sac, no? Because out of, see, megaspore mother cell, no. Okay, megasporophyll means the one that is going to keep arise. Megasporangium is not going to be, so it's going to be a megaspore. Okay, yes. Okay, chalo. The region where the body of the ovule fuses with the funicle, the region of where the body of the ovule fuses with the funicle is called raphe, hilum, tegmen, and caruncle. Which one? Yes, these are NEET level questions. Raphe, hilum, the region where the ovule fuses with the funicle. The region where the ovule fuses with the funicle. It's going to be the hilum, right? It's going to be the hilum. Very good, very good. Yes. So that's it for today, students. Yes, we are done with sexual reproduction and flowering plants. Next week we will do the next chapter, and today evening we'll be doing biotechnology principles, so definitely do not forget to join. And also Tanuja and Roshni, inform the other students in case for me travel takes a lot of time. I will be taking the class max by 4:30. Okay? Yes? Okay, chalo. So see you all in next class. Thank you so much for joining. See you all in next class. I hope all of you enjoyed the session as well as understood everything. So make sure that you learn this chapter. It's one of the most important chapters of your board as well as NEET, and it is a beautiful chapter. So try to learn the chapter and try to understand it very well. I hope. Thank you so much. Okay, for joining, and see you all.